Hello, ladies and gentlemen, and welcome to today's broadcast on developing multidimensional Skyline spectral libraries for rapid lipid analysis by Kaylee Kirkwood. Kaylee is a graduate student working in the Baker Laboratory in the Department of Chemistry at North Carolina State University. And I'm Susie Valdez of LabRoots, and I will be moderating today's event. Before we begin, I want to encourage you to engage with us. You can submit as many questions as you want at any time during the presentation. Just simply type them into the Ask a Question box on the left and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, just click on the Support tab found at the top right or use the Ask a Question box on the left. Please join me now in welcoming our presenter, Kaylee Kirkwood. Kaylee, you may begin your presentation.
Okay, good morning, and thank you all so much for tuning in today. I'm Kaylee Kirkwood, a second-year grad student in Dr. Aaron Baker's lab at North Carolina State University. And today I'm going to be telling you about some of the work that we've been doing to develop multidimensional spectral libraries for lipid data analysis using the free and open source software Skyline, which is out of the McCoss lab at the University of Washington. So here in the Baker lab, we collect lots of data for lots of different types of molecules, essentially anything that is measurable using mass spectrometry, including xenobiotics, proteins, glycans, lipids, and other metabolites. We then use that data to understand phenotypic outcomes for different conditions in various environmental and life sciences applications. And so my focus has been really on the data analysis aspect of this, and specifically for lipidomics data, so that we can confidently annotate our really complex data to gain some insight on how these lipids are actually changing in the different conditions that we're looking at. So to review lipids, for those who may need a little refresher, lipids have many important biological roles, including energy storage, signaling. They are structural components of cell membranes, and they're precursors in hormone biosynthesis. So here I'm showing the 8 lipid categories, which are defined by lipid maps. Each category has the common core structure that's shown on the slide. And so you can see that these lipids can be pretty structurally diverse. And then if we take a closer look at any one of these categories, we also will have multiple lipid classes. And so here are 10 lipid classes or head groups, in this case, all of which are part of the glycerophospholipid category. And so the ones that I'll be referring to the most in this presentation are going to be the PEs or phosphatidylethanolamines and PCs or phosphatidylcholines. And so here we have a lipid that we would denote PC 180181, where PC is going to refer to that phosphatidylcholine polar head group. And then 180 and 181 refer to the 2 nonpolar carbon chains, one of which has 18 carbons and no double bonds, and the other one has 18 carbons and 1 double bond or 1 degree of unsaturation. And so one of the most challenging aspects of lipid analysis is the complexity of the data, which is due to lipid isomers. And so here we have 2 lipids from different classes. One is from PE and the other one is PC. And these have different fatty acyl compositions, different head groups, but they end up with the same M/Z value with that M+H. And then we can also get isomers from a change in the fatty acyl tail composition. So here we have the same total number of carbons and the same total degrees of unsaturation, but the actual tails themselves are different, like this PC 180181 and then 16021. And in most cases, this is the level of speciation that we can achieve with our current technology and the workflow that we're using. And then another form of isomers comes from the change of the fatty acyl tail orientation, which is called the SN position. And this is where the fatty acyl tails are the same, but the connectivities are different. And finally, we can get lipid isomers based on changes of the double bond position. So here we have the double bond at the 9th carbon versus the 11th carbon. So to deal with these isomers, we use a liquid chromatography ion mobility mass spectrometry method to achieve multidimensional separations. And so we use a reverse phase C18 column to separate the lipids by their hydrophobicity. We use ion mobility, which I'll talk about in a second. And then we use a QTOF mass spectrometer in all ions mode, meaning that we collect our data with alternating frames of low and high collision energy to get precursor and fragment ions for the lipids. And so hopefully everyone is pretty comfortable with LC-MS, but I'm just going to give a brief explanation of ion mobility spectrometry, specifically the type that we're using in our lab for anyone who might be less familiar with that technique. So we use an Agilent 6560, which is a drift tube ion mobility QTOF instrument. The drift tube is filled with a buffer gas, such as nitrogen, and then ions pass through the drift tube, and those with the bigger size will have more collisions with that buffer gas, and so they take longer to travel through that drift tube. And so the output we get is going to be drift time, which is going to be larger for bigger molecules. These drift time values can easily be converted using what is called a single field calibration to collision cross sections, or CCS values, which describe the surface area of the molecule, and they are molecular descriptors, so they can be compared across labs and compared to database values. And so here, the more extended lipid is going to have a higher drift time and therefore a higher CCS value. And so while there are many benefits to using ion mobility, there are also challenges. And so the multidimensional data that we get is very large in complexity and file size, and manual assignment can be time consuming and can only be done for a few targets with small data sets. And so here we have an example of what that complex data might look like. And so we have a base peak chromatogram, which actually looks pretty clean compared to a lot of our lipid samples. And if we want to take a closer look at two of the peaks here, we can see the abundance maps, which show the drift time versus M/Z, with the abundance being shown as a heat map, where red is going to be our most abundant, and then blue is just going to be essentially noise. And so as you can see, each one of these peaks has a lot of features, so there's really a lot to tease out in this data. And we're also only looking at the low energy or precursor scans here. And so this can get even more complex when we start looking at those high energy fragmentation scans as well. And so to analyze this complex lipidomics data, there are currently two main options, which we like to say is sort of like a choose your fighter match between the manual labor and the black box. So the black box takes your data, compares it to database information, and gives you a list of lipids, usually with some sort of rank or confidence score associated with each identification. So the strength for this method is that it's extremely fast, it's good for large data sets, and it does a deep search through extensive databases. The weaknesses of this method are that there's little to no validation for the identifications. Lipids are often over-annotated, meaning someone may report the SN and double bond position of a lipid when they may only have data to support the fatty acyl composition. And then you may have to sacrifice some of that precious multidimensional data that you spent so long trying to collect, such as retention time and CCS, because some databases won't use those values. And then the search may not be sample or species specific. And on the other hand, the manual labor is where we started when I joined the lab, and that requires that you manually determine all the characteristics for your lipid of interest. So you have to find the formula, calculate the M/Z with the adducts you're interested in, find the fragmentation information, look in another database for the CCS value. So it can be very time consuming just to collect all of that information. And then you have to go dig through your data to find those lipids. And so the strengths of this method are that You get very confident annotations. You can utilize all of that multidimensional data, and you can prioritize biologically relevant lipids, meaning those that you expect to see in your species and sample type, or those that you're interested in given the specific conditions you're looking at. But the weaknesses of this method are that it's extremely time consuming and it's not sustainable for large data sets or deep searches. And so what we really wanted to do was take all these strengths and create our own method to analyze our lipid data. And so we decided that Skyline was the ideal software to do that with. So when we started using Skyline for our lipid data analysis, we started out with a really simple workflow, about as simple as you can get. So Skyline, for those who aren't familiar with it, is a targeted data analysis software, but it still works well for untargeted data as long as you have a list of molecules or M/Z values of interest to start with. And so our initial workflow started with just a simple list of lipids and their M/Z values, importing and analyzing the data, and then exporting a little bit of information. But since then, we've worked with the Skyline team to add some new small molecule capabilities, and then we've also taken advantage of a lot of their existing features. And we've come up with a proposed workflow for those who are interested in using Skyline for lipid analysis, where everything in yellow is new compared to our initial workflow. And so now our target list has lipids that have formulas, meaning we can look at the isotope distributions. We have multiple adducts, fragments, even some neutral losses, our CCS values, and normalized retention times. And now when we import our data, we're filtering in multiple dimensions instead of just by M/Z. And if we're using internal standards, we can also do some quantitation. And then we export a lot more information. And so some of the features that I'll talk more about in the rest of the presentation are spectral libraries, IRT retention time, and drift time filtering. So what we've been doing is taking our data from many different sample types and developing spectral libraries in Skyline, where each lipid has information on its M/Z value, its fragments, its CCS, and its retention time. And so the strengths of this method are that it is sample specific, multidimensional. The analyses are rapid, but you can manually validate your results. It's good for large data sets and target lists. Anyone who imports the spectral library can prioritize the lipids that they're interested in and can also customize the target list for their use. The libraries are generated from deep searches with confident and appropriate annotations, meaning they're not over-annotated. And Skyline offers straightforward visualization and quantitation tools, and of course is vendor independent and free. And so our resulting libraries contain hundreds of lipids from diverse classes from these 5 lipid categories out of the 8 defined by lipid maps that I mentioned earlier. And the other categories either aren't present in the samples that we've looked at so far, or they just require more specialized extractions. So to give you an example of what a single library might be comprised of, here we have a sample-specific library which has over 750 features from both positive and negative mode. And this boils down to about 500 unique lipids when you consolidate any lipids that have multiple adducts or that we're annotating in both ionization modes. And then when we plot this library, all the lipids here by their collision cross sections and M/Z values, we get these really nice trend lines for each lipid class. And so in positive mode, you can even see trends within the trend lines, and that corresponds to lipids that have the same total number of carbons, but varying degrees of unsaturation. And these trend lines can really help us validate our lipid annotations by verifying that we don't have any outliers, which would point to that lipid maybe belonging to a different class. And so this can be helpful when we're looking at those isomers that arise from those class or head group changes that I talked about earlier. And then we can also compare our experimentally determined CCS values with those present in the CCS compendium out of John McLean's lab at Vanderbilt for more confidence in our annotations. And so all the CCS values for the lipids in our current libraries that have a compendium match are less than 2% different than that database value, which is right where we'd want to be. So while our user workflow is really straightforward and only requires some manual work for validations, we put a lot of work into creating these libraries. So I wanted to just take a moment to go over that process for those who may want to create their own libraries for a unique sample type or maybe even a different set of molecules. So first we use the Lipid Creator tool in Skyline to generate transition lists with thousands of lipids. And this is another free tool. It's actually available as a plugin for Skyline to interface directly with the software, and I highly recommend it for anyone who is doing lipid work. We then search for those lipids in our data and find candidate peaks based on their mass measurement accuracy, the coelution of adducts, and the anticipated retention times given the lipid subclass. Then we use the Skyline Ion Mobility Predictor tool, which is used to calculate CCS values of the candidate lipids, which we can do with our single field calibrated data to convert any feature drift times to CCS values. And then lipids are annotated based on their drift time aligned fragments and their CCS values. And then for each library, we create a new IRT calculator and calibrate it to contain normalized retention times for all the lipids that are in our library. And I'll go into some more detail on that in a moment. And finally, we create and export our spectral libraries, and we plan to share them on Skyline's online data repository Panorama, where anyone can access and download them. So like I mentioned earlier, we use Skyline's indexed retention time, or IRT calculator, for retention time predictions and normalizations. And so to walk you through how we use this tool, we start by assigning a set of about 20 reference lipids that span our LC gradient, and then Skyline will assign them a relative IRT value from 0 to 100. And so we use endogenous lipids as our reference points to make sure that anyone can use it, but many people will use internal standards for this. And so we have plans to actually create secondary calculators for our different libraries using the new Avanti Ultimate Splash 1 mix, which is a mix of about 70 lipids from different classes. And so we can use that as a set of more standardized reference points. And so once we calibrate the calculator, we can add the rest of the lipids in our library so that each one is now assigned a unique IRT value. And Skyline recently added the option to use, in addition to linear regressions, either logarithmic or lowest regressions to set the IRT values. And so we've actually found that using a lowest regression has worked pretty well for us when we are looking at data from LC methods that have different gradient times, because linear regressions just don't always capture the differences where some gradient regions may be more compressed than others when you lower that time. And so both of these two steps are something that we handle while we're generating the library. And then someone who's using it would go in and use the IRT values to now predict the retention times of all the lipids in a new data set, regardless of the gradient time they're using or any retention time shifts. So a good example of the prediction potential of the IRT calculator is with lysolipids, which are lipids that have one fatty acyl chain cleaved off. And so the SN1 and SN2 lysolipids actually generally do separate by LC, but they have identical fragments, and then usually the CCS values are too close to distinguish them. And so it's important for us to know which peak is for which lipid. And so here, Skyline is correctly predicting that the SN2 lysolipid elutes first. And then using that auto-calculate regression feature, Skyline will actually adjust for any retention time shifts between samples, as you can see here with the two different predictions at 8.3 minutes versus 8.4 minutes when that peak shifts just a little bit between these two samples. And then we can see that the same thing holds true for the SN1 lysolipid, which is going to be that second eluting peak here at 8.7 or 8.8 minutes. And then in addition to predicting retention times, the IRT calculator is really useful for standardizing retention times. And so here we're looking at the retention time of a single PE across 4 different experiments. And this is in Avanti's brain total lipid extract, which is something that we use as a QC standard in all of our lipidomics experiments just to keep track of our instrument performance and our LC performance over time. And so this first set of 3 measurements was taken here at NC State on our current instrument, and then the rest were taken at PNNL on a different instrument and a couple different columns. And so you can see that this retention time can vary pretty significantly, anywhere from about 20 to close to 23 minutes. But when we convert to the IRT scale, which is just an arbitrary 0 to 100 scale, we can see that these values become much more comparable and standardized, all at about 79. So moving on from IRT, another feature of our workflow is spectral library matching, where you can view the library match just directly in that Skyline window next to your data, and you can make just direct comparisons to see how they match up. And so here we have an MS2 spectrum for a ceramide that we detected in one data set. And then here's the library match underneath. And then as you can see on the right side of the slide, these relative peak area profiles match up pretty well, so we can be confident in our detection of the ceramide in this experimental data set. And then another aspect of the libraries that I mentioned earlier is that each lipid has a stored CCS value, which can be used for drift time filtering if you're using ion mobility, but can easily be ignored if not. And so I mentioned that our CCS values are calculated from drift times using a single field calibration. And when your data file has been calibrated in the vendor software, which we do in our Agilent software, Skyline can actually back calculate drift time from CCS. And so we set a resolving power tolerance in Skyline just based on our instrument. And then Skyline uses both that CCS, the calibration, and then that tolerance to determine the window of drift times that contains the signal that we want to extract. And you can see that with the horizontal purple boxes here. And so in both of these examples, these other signals would interfere with our lipid of interest signal and probably contribute to the extracted intensities that we were getting. So we can try to filter them out based on their drift times to get a better idea of the true abundance of the lipid that we're actually interested in. And so this is a very important function, especially with lots of lipid isomers and lipids that differ just by one double bond. But I'd argue that drift time filtering can be even more valuable when it comes to lipid fragments, especially in negative mode. So as you can see with this PE lipid, the fragment signals for these 2 fatty acyl chains, 18,0 and 24, which are the teal and orange chromatogram traces here, pretty much dominate the extracted ion chromatogram. And this is due to the fact that most of the lipids in our sample with these 2 really common fatty acyl chains will share these same fragments. And our DIA method does not do any precursor isolation when we're fragmenting. So the same fragment can be seen along really wide ranges of our gradient. And so in some cases, these fragments can completely drown out the precursor, and this makes it difficult to annotate lipids and can throw off the relative intensities of our spectral library match. But if we use drift time filtering, we can filter out all of this excess signal from these shared fragments. And our chromatogram looks much more clear when only the signal that's contributing to our extracted intensity are at that correct drift time of our lipid of interest. So you can see at the bottom here at that fatty acyl 18,0 peak that we obviously had another lipid at that same retention time at a much higher drift time that was contributing to our fragment signal. And so one last capability that I'd like to point out, which can often get overlooked in lipidomics, is the utility of analyzing multiple adducts for a single lipid. And so in positive mode, PC lipids form really highly abundant protonated species, but collision-induced dissociation, which is the fragmentation cell that we're using, produces really low in abundance neutral loss fragments. And so in this example, which is another lysolipid, any pair of peaks here could be the correct pair if we were just looking at the protonated species, because those fragments are so low in abundance, we wouldn't really be able to tell them apart. But when we add in the sodiated species, which is this brown set of peaks, we can really clearly see which pair of peaks is the correct pair. So adding in that second adduct really helps us filter some of these candidate peaks. And then another example of where adding the sodiated species can be helpful is in assigning the fatty acyl chain composition of lipids. And so for this example lipid, which is pretty representative of most PCs, we can barely see the neutral loss fragments that would help us in the annotation of this lipid, even though it has a very high signal. And so we wouldn't really be able to call it a PC 18124 because we wouldn't be sure of those fatty acyl chains. And so here, the peak area of the protonated precursor is an order of magnitude greater than that of the sodiated species, but the sodiated species offers a lot more fragments to help in that fatty acyl assignment, and these fragments make up more of the total peak area with about 10% as opposed to about 1/4%. And so before I wrap things up, I just wanted to give some examples of different applications that our lab is working on that are using these libraries. So for example, I'm interested in assessing lipid changes in various systems following environmental exposures. So such as the ones on the top here, exposures to PFAS or exposures to combustion products in smoke. And so from these more environmental to some more life sciences applications, these lipid libraries are really facilitating a lot of the exciting research that's happening in our lab and hopefully in some other labs as soon as we make them publicly available. So in the meantime, if you're interested in using Skyline to process any lipidomics or other small molecule ion mobility data, or if you just want to learn more about creating and using spectral libraries in Skyline, we worked with the Skyline team to create a step-by-step tutorial for this type of data analysis. It's a hands-on tutorial with some of our fruit fly lipid extract data, and it should be posted on the Skyline website any day now. And so to wrap up, just a few take-home points would be that Skyline allows rapid and confident analysis of complex multidimensional data, which in our case is going to be LC-IMS-MS. And we're working to develop some sample-specific spectral libraries containing M/Z, retention time, and CCS data for hundreds of lipids. And then some of the key features of Skyline software, including IRT, spectral library matching, and drift time filtering, as well as seeing those adduct elution profiles, can really allow for some confident lipid annotations. And I'd also like to mention here that Skyline has lots of other features outside of what I've discussed today, and they're always adding in some new functionalities to their software to make it useful for really any data analysis workflow. So I highly recommend checking it out and seeing how it can fit into your data analysis workflow. And so with that, I would like to thank the Baker Lab members, our funding sources, specifically the NIH Molecular Biotechnology Training Program, and the whole team at Skyline, especially those who have worked on the tools that I talked about today or have worked on the small molecule support. And of course, thank you to the Agilent team for inviting me to speak today and to you all for listening. And with that, I'm happy to take any questions. I recommend checking it out.
Really for that incredible presentation. We will now begin the live Q&A portion of our presentation. Again, check out the left side of the Q&A box and ask a question. So we hope you will do that. We already have some great questions coming in. Our first question, what are some other ways Skyline can be incorporated into lipidomics data analysis workflows?
Yeah, that's a great question. And I think it's important to point out that what I was talking about in this presentation is outside of the norm for what people are using Skyline for. So Skyline, like I said in the presentation, is a targeted software. And so certainly any targeted lipidomics data, if you're doing SRM, MRM, PRM type methods, that is really what Skyline is built for, and it's a big workforce for targeted data. And so that's certainly a workflow where it's really easily incorporated. Even before you start running any samples, you can use it for your collision energy optimization, creating methods. And so that's definitely one place where it fits in well. And then another one would be with validating any results that you do get from any database searches. So if you're using a software like Lipid Annotator or Lipid Blast, one of the ones that are vendor neutral, then you can always sort of rework the spreadsheet that you get at the end there, and you can just throw that into Skyline and then just validate the peaks that you are seeing in the database.
Thank you so much. And do you know what the interferences could be on the drift time filtering slide?
Yeah, so on that slide, those data files were collected in negative mode. And so those were both PE lipids. And I believe those interferences were 1 Dalton lower than the lipids that we were looking at. So I would say that those were likely SM lipids, the sphingomyelins, because those are sort of in that same retention time window as the PE lipids, and just 1 Dalton away from the PE. If they were 2 Daltons away, I would say that that would be a loss of a double bond, which is pretty common in lipidomics analyses.
Thank you so much. And thank you to our audience members for these great questions. How much do you manually validate the peak picking in Skyline, Kaylee?
Yeah, that's another great question. And I would say that I try to manually validate all the peaks that Skyline has picked, even if it's just a really quick run through to check that the retention times match up and that the peak area profiles match up. I think that I guess I'll try to quote Will Thompson from Duke. He said that the same things are happening with any software that you're using, where you might get peaks that are picked improperly, but Skyline just gives you the opportunity to fix it. And so I definitely take advantage of that opportunity and fix any of the peak picking boundaries that kind of go awry.
Thank you so much. We have time for one more question. What is your typical LC-MS runtime?
So our typical runtime, we use a 38-minute gradient, which is pretty long. And I'll just wrap in another question that I see in the chat with that, which is that we've gone down to about 19 minutes. And that is kind of where we'll start losing some of the separations that we would get. And so really anywhere in that 20 to close to 40-minute range is where we're comfortable running our LC methods.
Thank you, Kaylee, for this informative presentation. Would you like to provide the audience with some closing remarks before we go today?
I'll just say again, thank you so much to the Agilent team for inviting me to speak here today, and thank you all for tuning in.
And thank you again. And as a reminder to our audience, questions we did not have time for today, and those that are were not addressed during the on-demand period will be addressed via your email you provided at the time of registration. And I want to thank our speaker again, Kaylee, and to everyone who participated in the presentation. We hope you will check out the exhibit hall and enjoy the rest of the Agilent event. Take care. Stay healthy. Bye-bye for now.